So we're going to continue on with chapter 21. The last talk was we started talking about the cellular immunity, and that's carried out by T cells. And then we were talking about the two different types, the CD8 and how they are going to differentiate into cytotoxic T cells, and CD4 are going to differentiate into helper T cells and regulatory T cells. And then they both can differentiate into memory cells. Now, helper T cells, it's listed at the bottom, but I feel it's really important to say. Without them, there is no immune response. They play this central role in the adaptive immune response. They activate both humoral and cellular immunity. And once they're primed by the presentation of the antigen by the antigen-presenting cells, the T cells will then, activate B cells and other T cells, induce T cell and B cell proliferation, and secrete cytokines that are going to recruit other immune cells. Now, the activation of B cells. So that's what we're talking about now, is how we're going to activate B cells with these helper T cells. And these helper T cells are going to interact directly, with B cells displaying antigen fragments bound to MHC2 receptors. They're going to stimulate the B cells to divide more rapidly and begin antibody formation. The B cells can be activated without helper T cells, possible, by binding to T cell independent antigens, but the response is weak and short-lived. And most antigens are T cell dependent antigens that require the helper T cell co-stimulation to activate the B cell. So again, this means that in order for most B cells to become activated, they're going to require the help of helper T cells. CD8 cells require helper T cells to become activated into destructive cytotoxic T cells. They also play a role in amplifying the innate defenses, and they activate macrophages, which is going to lead to more potent killers, and then mobilize lymphocytes and macrophages and attract other types of white blood cells. There are some subsets of Th cells, and I'm not going to ask you to memorize which number corresponds with which function. I would like you to know the functions in general, though. So Th1 cells mediate most aspects of cellular immunity. Th2 defend against parasitic worms, promote allergies. Th17 links the adaptive and innate immunity by releasing interleukin 17, and it may also play a role in autoimmune disease. And remember, autoimmune disease is where the body is attacking self-antigens. So now we're going to talk about cytotoxic T cells. And what they do is they directly attack and kill other cells. Activated cytotoxic T cells do circulate in both blood, lymph, and lymphoid organs. And they're in search of body cells that are displaying the antigen that they recognize. Remember, each cytotoxic T cell is going to recognize one specific antigen. So that cytotoxic T cell circulates around looking for that one specific antigen. Excuse me. Activated cytotoxic T cells will target cells that are infected with viruses, cells that have intracellular bacteria or parasites, cancer cells, and then foreign cells, like foreign cells obtained through like transfusions or transplants. Cytotoxic T cells deliver this lethal hit using two mechanisms. The first is they release these things called porphyrins and granzymes by exocytosis. And the porphyrins are going to create a pore where the granzymes can actually enter the target cell. And then the granzymes are going to stimulate apoptosis, which again is programmed cell death. And the cytotoxic T cell also binds to specific membrane receptors on target cells and stimulate apoptosis. So we have first cursor. We have one, the identification of the foreign antigen. This is what's occurring, well, we're trying to occur here. Two would be the release of porphyrin and granzyme, and that's what we're seeing here. 3, the porphyrin molecules insert, and that's actually what we're starting to see here. They're inserting into the membrane, but we definitely see it here, and it's going to form this pore. 4, the granzymes are going to enter through that pore, and they're going to trigger apoptosis. And then 5, the cytotoxic T cell detaches and goes in search for another prey. This is a cytotoxic T cell attacking a cancer cell. And then we also have natural killer cells, and they recognize other signs of abnormality that cytotoxic T cells do not look for, such as cells that lack the class 1 MHC protein, antibodies coating the target cell, and then different surface markers seen on stress cells. And natural killer cells use the same key mechanisms as cytotoxic T cells for killing their target cells. And then we have this immune surveillance. And this is where natural killer cells and cytotoxic T cells prowl through the body looking for markers that they recognize. T regulatory cells dampen the immune response by direct contact or by secreting inhibitory cytokines such as interleukin 10 and transforming growth factor beta. Important in preventing autoimmune reactions. Remember, again, autoimmune is when the body is attacking itself. It suppresses self-reactive lymphocytes in the periphery. And then there's some research into using them to induce tolerance to transplanted tissue. And this is a really nice table showing the different types of cells and molecules and their function in the immune response. And this is your question. That's all I have for you for this talk. I hope you have a wonderful day.